Yeah, so I want to thank the organizers for inviting me. It's definitely humbling for me to be here talking to this group, in part because of a disclaimer I want to give at the top here, which is that I don't do sleep research. But I'll kind of explain, hopefully, a little bit, excuse me, of why I'm here and I think why I was invited, with a little bit of my background. So just really quickly, I want to say that this is going to be an outsider's perspective on what I think are some of the big questions, in particular in the human sleep neurophysiology literature, which I'll explain to you kind of why I have some familiarity with that. And ultimately, my goal here is to see if I can give some possible novel approaches to try to answer some of these questions. I also just want to say, most of you are likely familiar with most of the methods that I'm going to talk about. And in fact, there are some people in the room who have used these methods for studying sleep to, in some really beautiful studies. But I think that as a field, you may not be as aware of some of what I'm referring to as the untapped potential of the kind of data that we have access to. And the short version is that there actually already exists a trove of data of humans sleeping that's really just waiting to be explored by what I think is going to require a fairly multidisciplinary team. So one of my goals here is to, if there's anybody who's interested in collaborating on this, I think there are some fairly obvious things to do. So just really quickly, in like 1 minute, I want to give you a sense of what my background in sleep is. I'm actually, as Katya said, I'm a cognitive and systems neuroscientist. My focus is on speech and language, and more recently on learning, using direct human neurophysiology methods, both recording and stimulation approaches. So I guess to some degree, my interests have clearly gone to the extremes of like the most human-specific behaviors like language and the probably least human-specific behaviors like sleep. But one of the reasons that I've been interested in sleep for a long time is that I actually did my PhD in a lab at UCSD that does study sleep neurophysiology in humans. And so I was kind of exposed to this literature, and over the years I've kept up with it just because I think it's really interesting. And I've been sort of taking notes on things that I've noticed in the literature and things that, if I ever had the time, I would want to do. And it turned out that actually in January 2018, through a long chain of events that I won't go into, DARPA actually recruited me to be a program manager. And mostly what I was going to go there to do is to do programs on things related to my main research, which is human communication. But they also, through some conversations with the folks at DARPA, we were talking about some other DARPA hard problems, and the one that really came up as like something that just needs to be done is sleep enhancement. And obviously the DoD implications of that are obvious, and then there seem to be some recent advances in this community in particular that might have made this a good time to do that. So in the time where I accepted that position, I spent about 3 or 4 months really trying to do a deep dive on the human sleep neurophysiology literature, try to understand it, try to identify what are the big missing pieces, and in particular, what can the methods that I'm familiar with contribute, in particular, the fact that we can do this in humans, things like potential control algorithms and closed-loop neuromodulation, and then also the application of multimodal methods, so how could we combine things like pharmacology and neurophysiology. And then in June, my security clearance was denied. The asterisk there says, for reasons that are not as interesting as what you're probably thinking, but very illustrative of the way that our government is dysfunctional. And you can ask me about that later if you want. So I did all of this work to try to get myself into this field, and kind of the outlet for it sort of fell out beneath me. But I'm still interested in trying to do this. So I've been thinking a lot over the last couple of years about what I can do, if not funding other people to do this kind of work, to do it myself. So again, I'm not going to talk about any specific data that we have analyzed in this domain, but I want to kind of just put out there what exists and what we could potentially do. So everything that I'm going to talk about is done in the context of epilepsy surgery. This is Eddie Chang, he was my postdoc mentor, and now for the last several years, very close collaborator. He's a functional neurosurgeon at UCSF and specializes in, among other things, epilepsy, his clinical practice. Again, most of you are probably familiar with this, but I'm just going to kind of go over like where these methods fit into the suite of tools that we have available to study the brain in humans. I'm sure you've all seen a version of this figure before, which is sort of mapping out the spatial resolution versus temporal resolution of these various methods. Some of the most commonly used ones really do kind of have this trade-off between these two dimensions, where functional MRI, which is really, I think, the workhorse of cognitive neuroscience, over the last 30 years, has pretty good spatial resolution at least at the map level. But pretty poor temporal resolution because it's measuring hemodynamic signals. And kind of conversely, EEG and MEG, the spatial resolution is not very good, even though it has pretty good temporal resolution. The methods that we work with, which here I'm referring to as micro-ECOG or microelectrocorticography, the goal is to try to get closer and closer to the bottom left of this graph. And we're sort of moving there through a variety of collaborations we have with some of our engineering collaborators. This has been the workhorse of most of what we've been doing over the last 10 years, which is these 4mm arrays of 256 electrodes. You can see they give us very good coverage for the kinds of things that we study with speech and language. But as I'll show you in a second, actually, when you combine this with some other methods, this is potentially very useful for answering some questions about the cortical neurophysiology of sleep. We're currently also working with collaborators at Lawrence Livermore National Lab to develop even higher density arrays that we can really push this basically as high density as we want to go. We need to identify what we think is the right target for that. So to give a little context of how this type of research, the context in which this type of research takes place, the two primary methods are electrocorticography, which is the surface recordings, and stereotactic EEG, where you place depth electrodes. And the basic idea is, at least the way that it's done typically at UCSF in epilepsy, is that there's a craniotomy to expose the brain surface, place these electrodes on the cortical surface. We also get pretty good coverage using high density grids and depth electrodes, as well as some linear strips of a variety of areas, including some of the mesial structures like hippocampus, amygdala, anterior, posterior cingulate, prefrontal cortex, orbital frontal cortex. And then, of course, we can do whatever we want to do with these signals to look at how they relate to behavior. So these kinds of methods have been used, I think, very, very fruitfully for a long time, actually, to study some really important behaviors in sleep in humans. And one thing I just want to point out is I think that most of this stuff has been done and I think can be done with fairly limited data. So just to give some examples very, very quickly, I'm not going to go into any details about this, identifying sort of what the multi-scale generators of slow waves are. This is a really, really nice paper showing basically all the way from scalp EEG through these depth electrode EEG recordings down to single units, actually what the various characteristics of slow waves are. This is a really nice paper that came out a couple years ago that actually used these thumbtack electrodes that penetrate the cortical surface and go down through the lamina, and looking at the generators of spindles and finding that there are both superficial and deep generators that are somewhat independent. Ripples also, of course, as this relates to things like memory and learning, the ability to look at the focal generators of those. And in somewhat rarer cases, given the fact that the coverage of where the electrodes are placed is always determined by the clinical needs of the individual patient, sometimes you actually do get coverage of some of the thalamic-cortical networks that are important for sleep. So all of this work is really done in this context where we have patients who are in the hospital. It's an inpatient procedure. They have these electrodes in their heads. They're waiting to have seizures. They usually sit there for about a week. And then once they have the seizures, those areas are identified and, if possible, safely removed, resected in a second surgery. But it's in that intervening time where we can do these recordings, we can ask patients to do tasks, we can ask them to collaborate with us to study a whole wide range of questions. Now, I wanted to just do kind of like a back-of-the-envelope calculation to give you a sense of the amount of data, the scale of data that honestly already exists and is sitting on servers. So at UCSF, we have roughly over the last 5 or so years, we average about 20 patients a year who get this kind of surgery. Each patient, an average of about 7 days in the epilepsy unit, and that's being recorded 24 hours a day. And then this is, of course, highly variable. It depends on a lot of things like pain tolerance and also the type of epilepsy they have. But let's just say they have about 6 hours of decent sleep a day. It may not be contiguous, but it's there. That right there is about 840 hours of continuous data, of sleep data. This is kind of what I mean when I say that there's this trove of data. It's literally sitting on our servers at UCSF waiting to be analyzed. And I'll talk in a minute about what I think some of the sort of low-hanging fruit questions might be. Although, of course, I'm very interested to hear what this community thinks about what those questions are. If we scale this up a little bit, I just threw this together. This is 45 random patients. Almost 13,000 electrodes, just to show you the scope of the coverage that we have on these large-scale data sets. I planned to plot 200 patients, but then I got to 45 and it wasn't showing up anymore because it's basically too dense. But you can see that it's definitely somewhat biased in terms of the exact areas that we get. Again, this is driven by the clinical necessity. But we get really nice coverage of these lateral areas, extremely dense coverage using the kinds of grids that we use. That can be interesting for things like traveling waves, and that's been used by several groups now. But we also get a fairly dense coverage of a lot of the prefrontal and medial structures that might be important for both sleep and the behavioral correlates of sleep. So now we can kind of start to think of sleep as a big data problem. And in the human intracranial field, there's this interesting paradox, which is that the data themselves we think of as being very rare, precious, and limited because it's only 20 patients a year at our center. Some centers it's even fewer. But it's a fairly small segment of the population that needs this kind of surgery. But at the same time, a given data set is really a lot richer than we actually use it for. Our typical data in the kinds of tasks that we do in our lab, and this is true across most labs that use these type of data, is you might record for 5 to 10 minutes while the patient's doing a task, and then basically throw out everything else. It doesn't get used for anything. And as I said, there are really hundreds or even maybe thousands of these data sets already available, let alone what we can continue to collect and augment with various other tools. So what are some of the big questions that we might try to ask with this type of data? The one that really stood out to me in reading the literature was this question about state transitions, and why we transition to various stages of sleep when we do. And in particular, this might be an interesting question to ask regarding the cortical generators of some of these things like slow waves and spindles, but also the ability to look across both short and long timescales. If we can look over 24-hour recordings, or at least several hours of continuous sleep, you can look at that, but you can also look at how that relates to some of these other more local phenomena that have been studied with these methods like spindles and K-complexes and things like that. Also looking at things like long-distance communication and coordination, traveling waves. A lot of these patients are already doing behavioral tasks that relate to learning, so we can look at the relationship to ripples. And then there are also some more sort of maybe translational questions that can be asked, in particular related to the relationship between sleep and epilepsy, which is interesting in and of itself because seizures do have several timescales of rhythms in which they occur, including multi-day timescales. And then, the potential for doing trials. If you have drugs that you think might be important for influencing or augmenting sleep, assuming that they can be done safely in this population, having access to direct neurophysiology data while you do that is obviously very valuable. And to do these kinds of things, we can obviously take advantage of the sort of revolution that's going on right now in deep learning, this has a lot of advantages, and I think that there are some fairly nice algorithms that exist for doing things like automated sleep staging, although that's by no means a solved problem at this point, particularly for this kind of data. But I also want to point out that when we're talking about these kinds of analytic tools, there's a lot of other data sources that we can take advantage of. So it's trivial for us to ask patients to wear monitors on their wrists or put them underneath the mattresses in the hospital beds so that we can try to get these additional biomarkers of sleep and in particular what sleep stages they're in, even if it's somewhat rough. But actually also because this is a clinical setting, we do have, this is just a screenshot of 24-hour audio and video. Patients they have a camera trained on them. They consent to this because it's useful for identifying behavior that's related to their epilepsy. This can be mined using both manual as well as some of the deep neural networks that have been trained, like convolutional neural networks that are very good at doing things like object detection in video. Plus, all of the other sorts of clinical sensors that they have. So most of these patients also have EKG, sometimes multi-lead EKG if that's something that's useful, as well as scalp EEG. So there's a lot of things here that are available. And then this also leads to kind of the other side of the analytic tool suite that's available, which is more unsupervised learning. So I think this is something that I haven't really seen in the literature. Maybe somebody has done this, but I think maybe not on this scale of having this amount of data just to be able to identify, how well do the sort of classic models of sleep states, for example, fit into what the data actually show. So I just want to, in the last couple minutes, talk about two other opportunities that are possible with these types of patients. One is neuromodulation. So most patients undergo various types of electrical stimulation for clinical mapping anyway as part of their treatment. We're trying to identify areas that are involved in motor behaviors and speech and language behaviors so that the surgeons know not to cut those out. And the research opportunities for doing stimulation are incredibly abundant. This is just a small list of recent studies, but this is a really growing field. And in particular, the use of closed-loop algorithms for influencing behavior, including at the level of even things like neuropsychiatric disorders. So there's some really interesting work happening at UCSF, among other places, to do that. So yeah, I mean, you can imagine various scenarios. This is just a schematic where it might be interesting to try, and people have done versions of this, of course, using optogenetics, as well as some non-invasive stimulation in humans to try to induce sleep rhythms by triggering the brain's sort of intrinsic sleep mechanisms. It remains to be seen whether this can be done non-invasively in humans. So the ability to stimulate directly from all of these implanted electrodes might actually be necessary. And then the last thing I want to just sort of end with here is, so everything I've talked about up to this point has been the case of these epilepsy patients who are sitting in a hospital bed. They are literally tethered to the wall. They have wires coming out of their heads, and they can basically go from the bed to the chair to the bathroom and back. But the last several years, we're starting to now have the ability to do this in a chronic setting. And maybe I should put quotes around ambulatory because people probably aren't walking while they're sleeping. But now there are several techniques available to do this kind of stuff, this direct neurophysiology in humans who have chronic implants. So, for example, right now there are about 160,000 patients who have DBS implants. Most of these are patients who have motor disorders like Parkinson's disease, essential tremor, dystonia. But as I mentioned before, this is also now being applied much more widely to things like neuropsychiatric disorders, major depressive disorder, OCD. There's trials for chronic pain now, the tinnitus. The list kind of goes on and grows every day. And the number of patients who have this is growing as well. And including work from Phil Starr's group at UCSF now, this technique is being augmented with the ability to place electrodes in other areas as well that might be useful for example, doing closed loop stimulation for Parkinson's. So the coverage is getting wider and wider in these particular patient populations. And then one other way in which this is done is there's actually now more than 1,000 patients, I think it's maybe even 2,000 patients, who've been implanted with chronic ECOG and stimulation from a device from a company called Neuropace, which is just down in the peninsula. This was sort of the first fully closed loop implantable device for humans that was approved by the FDA about 7 years ago. And yeah, I mean, in all of these cases, these are still fairly limited. We're talking a small channel count, relatively small recording durations and sampling rates, but low sampling rates. But actually, there's now been a bunch of work that's been published showing that these are really useful research tools. And if you can, it's very easy to think about ways that they could be applied in the domain of sleep. So yeah, thank you.
Perfect timing.
Thank you so much for sharing. A stupid question. It's just like in the surgery,
at times, I guess, you damage some tissue or you remove. What would be interesting is not only to record before, but after that. We know that circadian regulation of neurogenesis is important. The sleep-dependent part of it is not well known. It's true for neurogenesis and angiogenesis. So it would be nice to see that during the healing process in the regions around, the damaged tissue, if you have, like synchronicity or silencing, or of course, if at some point there is correlation, you could stimulate it.
Yes.
Just would that be, a good, system to, investigate in your setup?
Yeah, absolutely. And actually that sort of brings up a whole other set of possibilities here, which is that because many of these patients, not all of them, but many of these patients are getting a resective surgery as their sort of terminal surgery, we also have access to human tissue samples and being able to do single cell RNA sequencing and other using genetic tools to actually be able to look at the anatomical properties of the tissue where we have this high density physiology already recorded. That's another possibility. The challenges, I think, with doing sort of post-surgical or post-resection studies is that they no longer have the electrodes in after the surgery, after the resection surgery. But like I said, a lot of these patients, some patients actually do receive some amount of resection plus the chronic device implants after the fact. So that's certainly a possibility.
Just by curiosity, there is quite a bit. I mean, seizure is very complex, but there are a few seizure types. For example, frontal lobe epilepsies that are, specific during sleep, and we really don't understand very much the relationship between seizures and sleep. Mm-hmm. That is another area where I guess you could really get information about, we know sleep deprivation is supposedly increase seizures, but this is not even well demonstrated. And I'm just wondering if you have looked a little bit in that area. What will be the mechanism by which sleep and epilepsy interact?
Yeah.
Different type of epilepsy, which are actually being implanted.
Yeah. Well, so one thing to note just quickly is that there definitely is going to be some selection bias in terms of the types of epilepsy that these patients are going to have given the type of treatment. But at the same time, actually, there was a paper from Vikram Rao, who's a neurologist, an epileptologist at UCSF that came out a couple years ago, where they had access to, I think it was a couple hundred patients of people who had the NeuroPace implant. And what they were looking at was over very long time scales, what are the sort of rhythms of seizure activity? They did not break that down, as far as I know, by seizure type and by some of these other characteristics. And so that data, that's another data set that actually already exists, and you can work with the company to get access to it. And they're very, I think, very interested in collaborating with researchers for these kinds of questions.